Assalamualaikum warahmatullahi wabarakatuh and good day to the respected evaluators for today. My name is AMIRA from Group ES25760 and today I will be presenting on my final year project entitled ANTIBIOFILM of ACTINOMYCETES extracts against Salmonella typhimurium. Okay, bacteria can be in a form of planktonic cell or in biofilm communities that surrounded by extracellular polymerase substances, EPS, and can be found attached on surfaces. Due to the adverse environmental stimuli, for instance, changes in pH, oxygen, temperature, as well as nutrient availability, bacteria will form biofilm in order to seek protection. Biofilm forming bacteria can cause diseases which are considered difficult to treat due to its resistance towards treatment. On the other hand, Salmonella is a gram-negative and roach-in-shaped bacteria known to be the cause of foodborne outbreak, which has become the major concern of public health. These bacteria acquire the ability to form biofilm and attach on various surfaces such as plastic, stainless steel, and rubber. Next, actinomycetes produce the bioactive secondary metabolites that are known to have antimicrobial properties against various pathogens such as E. coli, Bacillus subtilis, and Klebsiella pneumonia. Actinomycetes are categorized in gram-positive unicellular filamentous bacteria and can be found in sea and sand sediments. The ability of Salmonella typhimurium to form biofilm which inherent resilience to the treatment is quite concerning, and antibiofilm agent has huge potential to lessen the infection globally. Therefore, as it is suggested, actinomycetes extract has enormous potential as new agent of antibiofilm. This study also provides the understanding on how actinomycetes extract controls the formation of biofilm. Community will be aware of the new finding on bioactive compound produced by actinomycetes as preventive measure against the biofilm-related infection of Salmonella typhimurium. And the objective of this study is to compare the antibiofilm activity of actinomycetes extracts against biofilm of Salmonella typhimurium. Biofilm formation consists of 4 stages. The first one is the attachment of planktonic cell on surfaces, formation of multilayer cluster, maturation, and dispersion. Biofilm acquire virulent mechanisms such as EPS that function in preventing drug penetration. Not only that, efflux proteins in efflux pump system contribute to the virulence of organism. Besides, polymicrobial biofilm interaction lead to the gene transfer between non-resistant to the resistant bacteria, which lead to the harmful outcomes. Biofilm-related bacteria can lead to the various infections such as urinary tract infection, gastroenteritis, sinusitis, and many more. Okay, next, actinomycetes produce bioactive secondary metabolites, which is often due to depletion of essential nutrients. Most antibiotics are derived from actinomycetes. This bioactive compound possess antimicrobial properties against various pathogens such as E. coli and Proteus vulgaris. Okay, method that were carried out on this research are confirmation tests on Salmonella typhimurium isolate ATCC 14028 by gram staining, catalyst test, and Pilka assay. The inoculum and treatment were loaded in 96 well microplate and incubated at 3 different incubation periods, and crystal violet assay was performed for the observation. Isolates that received from the laboratory was confirmed to be Salmonella typhimurium. The inhibitory effect of actinomycetes extract on Salmonella typhimurium biofilm were further studied. 4 species from actinomycetes isolate, namely S5, S7, AA141, and A11 that isolated from the brisol and obtained from UPSI were used as treatment for biofilm. They were observed at 6 hours, 12 hours, and 18 hours of incubation period, and extract concentration used were from 10% to the 50%. Okay, ethyl acetate S5 was also first, and based on the result obtained at 6 hours, showed no inhibition at 50%, 40%, 30%, and 10% of treatment concentration, while at 12 hours revealed the inhibition at all concentration except 40%, while 18 hours showed no inhibition except 50 and 40%. Next, ethyl acetate S7 was observed 50 and 20% were the only concentration that showed no inhibitory effect at 6 hours, while at 12 hours it demonstrated sign of inhibition at all level of concentration, and unfortunately at 18 hours no inhibition occurred except at 10% of concentration. The third observation was on ethyl acetate AA141, showed no inhibition at 6 hours and 18 hours except 10% concentration in 6 hours incubation time, while 12 hours revealed the positive inhibitory effect of actinomycetes extract on biofilm. Okay, lastly, ethyl acetate A11 also shows sign of inhibition at all level of concentration during a 12 hours incubation period. Meanwhile, 6 and 18 hours show no inhibition except at certain level of concentration. Therefore, Based on the data above, it shows the inconsistency of inhibitory effect of ethyl acetic extract towards the biofilm of Salmonella. 12 hours show the greater inhibitory effect. This might be due to the susceptibility of young biofilm toward the antimicrobial compound. No inhibition at 18 hours can be inferred from the EPS metric that prevent the entrance of antibiotic. Salmonella typhimurium also possess ACRB protein that somehow can be overexpressed and reduce the susceptibility of bacteria itself. To conclude, ACTINOMYCETES extracts show poor inhibitory effect on Salmonella typhimurium biofilm. However, this new finding of bioactive compound as antibiofilm could be used for the future reference in identifying new novel drug against biofilm related disease. Chemical analysis is highly recommended for better understanding in regards to bioactive compound composition, and the extract could possibly be combined with certain inhibitor and autoinducer to target certain virulent gene to inhibit biofilm. So I think that's all from me. Thank you.